So today I'm going to discuss with you all about the mitochondrial biogenesis that is given in your cell biology syllabus of BT first sem. So previously I have done about the functions of mitochondria and the structure of mitochondria. Just go through these lectures and let's get started now. So about the mitochondrial biogenesis. What is mitochondrial biogenesis? Mitochondrial biogenesis is the process by which your cell increase the mitochondrial numbers. So it is a process by which your number of mitochondria get increase. And this process was first described by John Hollows Z in the year of 1960s. And it was discovered that due to physical endurance training, it induces higher mitochondrial content levels, leading to greater glucose uptake by your muscle. Physical endurance means physical exercise. As you will see in these following notes, I have given the word meanings of physical endurance. So, due to physical endurance training, John Hollows Z have discovered that due to physical endurance training, it leads to high mitochondrial content levels, and that leads to greater glucose uptake by our muscle cells. Mitochondrial biogenesis is activated by numerous different signal mechanisms during times of cellular stress or in response to some environmental stimuli like your exercise, especially aerobic exercise that we normally do in the gym. Okay, so it is a process. So the thing that we are going to discuss here is about the biogenesis, where how we can increase the number of mitochondria within our cells. And it is saying by John Hollows Z that due to physical endurance like aerobic exercise training, it causes to induce high level of mitochondria, right? And the process of mitochondrial synthesis is actually activated by numerous different signal mechanism. Different genes regulate the process of synthesis of mitochondria. Well, as I have said here, that what is aerobic? Aerobic exercise means what? It refers to as relating to involving or requiring free oxygen. And simply aerobic exercise means use of oxygen to meet the energy demands during exercise. Well, during exercise we need oxygen, and that's why the name comes aerobic exercise. And during this process we need lots and lots of oxygen, and by the process of aerobic metabolism we try to burn our fat, trying to burn our the excess glucose which is stored in the form of glycogen. And during this process, as mentioned by John Hollows Z, that your mitochondrial biosynthesis occurs. Now, the ability for a mitochondrion to self replicate is rooted in its evolutionary history. Now, we have said, according to the endosymbiosis theory, that how mitochondria self replicate, and that's the main root for its evolutionary history, because mitochondria have their own genome. Although most of the some of the mitochondrial proteins are synthesized from the nucleus and then they are transported into the mitochondria by the help of channel proteins. So, by the help of translocators, you can say also. So, the ability of the mitochondrion to self replicate is rooted in its evolutionary history. It was commonly thought that mitochondria have come from the cells that forms a endosymbiotic relationship with the alpha proteobacteria. And so it is saying that mitochondria have come from alpha proteobacteria, which have form a mutual, you can say mutual relationship with the eukaryotic cells and remain within the cells, as mitochondria have their own genome for replication. This was about the endosymbiotic theory. Generally, endosymbiotic or endosymbiont means an organism which lives within the body of or cells of another organism, most often not always in a mutualistic relationship. So, here the mitochondria, as stated by the endosymbiotic theory, they have come from alpha proteobacteria and they remain within the cells of eukaryotic origin. They have said because the provement is mitochondria have their own genome, as you can see. But some of the proteins of mitochondria are coming from the nucleus, from the nuclear genome. So, however, recent evidence have suggested that mitochondria may have evolved without the symbiosis. This is a bit of a debate, you can say, that most of the case we say that mitochondria was once upon a time it was a organism of alpha proteobacteria origin, its ancestor, and have according to the endosymbiotic theory it forms a endosymbiotic relationship with the eukaryotic cell. But recent evidence has been found that mitochondria may have evolved without symbiosis because some scientists have observed that some of the mitochondrial proteins are synthesized by the nuclear genome, which are transported after translation. They are transported into the mitochondria, right? The mitochondrion is a key regulator of the metabolic activity of the cell, as we know the electron transport chain, the oxidative phosphorylation, okay, the TCA cycle, the main events of the energy metabolism takes place within the mitochondria. So it is a key regulator, important regulator of the metabolic activity of the cell and is also an important organelle in both production and degradation of free radicals. As we know, free radical can damage to our cells from the reactive oxygen species. So it has variety of functions. So it is an important organelle. It is postulated that high mitochondrial copy numbers or high mitochondrial mass is protective for the cell. So according to the biogenesis of mitochondria, it is saying that high mitochondrial copy or higher mitochondrial mass is required by the cell for its protection. Okay. Now, as I have mentioned that some of the proteins are synthesized from the nucleus, some of the proteins of the mitochondria are synthesized within the nuclear genome. So mitochondria not do not have all of its proteins, all of its gene encoding proteins. So mitochondria are produced from transcription and translation of the genes of both in the nuclear genome and in the mitochondrial genome. Mitochondria have their own genes. Okay. But some of the proteins that are found in mitochondria, they have been translated from the nuclear genome, right? Okay. So the majority of the mitochondrial protein comes from the nuclear genome as stated in the theory, which is totally different from the endosymbiotic theory. Okay, that most of the mitochondrial protein comes from the nuclear genome, while the mitochondrial genome encodes some part of the electron transport chain along with the mitochondrial RNA and tRNA. So, as stated to the biogenesis of mitochondria, majority or most of the mitochondrial protein comes from the nuclear genome, while the mitochondrial genome encodes some parts of the electron transport chain along with mitochondrial RNA and tRNA. Okay, so most of the proteins come from the nuclear genome, as stated by the scientists. According to the when we are talking about the biogenesis of mitochondria, mitochondrial biogenesis increases the metabolic enzymes for glycolysis, oxidative phosphorylation, and ultimately a greater mitochondrial metabolic capacity. So, if the number of mitochondria get increased in the cell, then obviously it will increase the enzymes, increase the metabolic enzymes so that they can catalyze the glycolysis process more efficiently, more fastly, the oxidative phosphorylation process, and ultimately greater mitochondrial metabolic capacity. However, depending on the energy substrates as available and the reduction oxidation state, which is redox state of the cell may increase or decrease in the number and size of the mitochondria. Now, depending on at what state the cell is present, okay, the redox state of the cell, that is the reduction of oxidation state of the cell, the energy substrates are available or not, or how much is available, okay, that can leads to the cell to increase or decrease the number and size of the mitochondria. Critically, mitochondrial numbers and morphology may vary according to the cell type, according to the context specific demand, where the balance between mitochondrial fusion and fission regulates the mitochondrial distribution, morphology, and function. Now, there will be a fusion or fission. Fission means to break apart the mitochondria into small mitochondria, and fusion of the mitochondria may occur. It depend on what type of cell we are observing, depending on the context specific demand that we require high amount of mitochondria or not. So increasing the mitochondrial biomass can be observed depending on the morphology of the cell, depending on the what type of cell we are using, what type of cell we are observing. Okay, and there must be a balance between the mitochondrial fusion and fission products. Obviously, this fusion and fission is regulated by some genes that we will discuss now. So this is a image where we can see mitochondrial proteins are encoded from the nuclear genome, and they are need to be targeted and transported appropriately into the mitochondria. These are translocators where they allow the transport of these proteins. These are actually needed for the mitochondria process to occur. Okay, and they are being transported from the outer membrane, they are entering to the inner membrane with the help of these translocators. Okay, okay. So since majority of the mitochondrial proteins come from the nuclear genome, the proteins need to be properly targeted and transported into the mitochondria to perform their functions, because there are inner membrane and outer membrane and they have particular functions within the mitochondria, right? So proper targeting of the proteins must has to be done, because as I have said that majority of the mitochondrial proteins come from the nuclear genome. So the proteins, The mitochondrial proteins need to be properly targeted and transported into the mitochondria to perform their functions. The first step for protein targeting into mitochondria is the mRNA, which is being translated within the cytosol of the cell. Then the resulting unfolded precursor protein will be able to reach their respective mitochondrial compartments, that is to the inner mitochondrial membrane and to the outer mitochondrial membrane. But they are unfolded, right? They have not been folded properly, and folding of the protein must be required for their appropriate function and the stability of the protein. Precursor proteins will be transported to one of the four areas of mitochondria, which includes the outer membrane, the inner membrane, the intermembrane space, and matrix. So there are four locations. There are four locations of the mitochondria where the precursor protein, the unfolded proteins, has need to be transported. Either depending on where they will act, depending on where the protein will be acting, they are doing their function. To the outer membrane, the outer mitochondrial membrane, to the inner mitochondrial membrane, to the intermembrane space, and to the matrix. Now, generally, all the precursor proteins, that means the unfolded proteins, which has been recently being synthesized in the cytoplasm of the cell, these mitochondrial precursor proteins first enter to the mitochondria with the help of a translocase on the outer mitochondrial membrane. So, as given in the diagram, uh, we will see that all the proteins enter to the mitochondria. All the proteins, all the mitochondrial precursor proteins which has not been folded now, they are entering to the mitochondria by the help of a translocase protein found on the outer mitochondrial membrane. Now, the name of the translocase is TOM. So, with the help of translocase, all the precursor mitochondrial protein are entering. So, from the outer membrane, it enters. Now, some proteins will have an N-terminal targeting signal, and these proteins will be detected and transported to the matrix. Now, there are some mitochondrial precursor proteins which have N-terminal, means amino terminal targeting signal. Those proteins, those mitochondrial precursor protein that have an N-terminal targeting signal, these proteins will be detected by the mitochondria and will be transported into the matrix, that means at the innermost part of the mitochondria, where they will be then cleaved. Their N-terminal signal sequence will be cleaved, that means they will be cut down. The signal sequence will be cut down, the N-terminal signal sequence will be cut down after reaching to that matrix site, and then they will be folded properly. So, those mitochondrial precursor protein that have an N-terminal targeting signal, they are destined and they are detected and they are transported within the matrix, that means innermost part of the mitochondria, where they will be cleaved. At what region they will cleave? The N-terminal targeting signal will be cleaved now, and then they will be folded. Other proteins may have targeting information in their sequences and will not include an N-terminal signal. So, there are also some other proteins, mitochondrial precursor protein, which are destined to be embedded in the inner mitochondrial membrane or to the outer mitochondrial membrane, but they do not have that N-terminal signal sequence, but they have a specific sequence targeting information in their sequence, in their amino acid sequence, but not N-terminal signal sequence. Okay, the N-terminal signal sequence precursor um, mitochondrial proteins are destined to enter to the matrix. where they will be cleaved and folded. But the other mitochondrial proteins, they have their targeting information within their amino acid sequence, and they will be destined in their particular location where they will be doing their function. Okay, so I think you have understood that all the mitochondrial precursor proteins enter to the mitochondria by the help of a translocase known as TOM. There are some mitochondrial precursor proteins which have a N-terminal targeting signal. These proteins will be targeted and transported to the matrix of the mitochondria. After entering to the matrix of the mitochondria, the N-terminal targeting signal will be cleaved and it will be folded, because the that proteins has not been folded properly. Na? It has to be folded for its stability and to perform its function. Some other mitochondrial proteins, they have targeting sequences, they have targeting information within their amino acid sequences, but not they don't have n terminal targeting sequence. So these other proteins are destined to be transported to their respective locations within the mitochondrial membrane, either in the inner membrane or to the outer membrane. Okay. During the past 2 decades, researchers have discovered over 30 proteins that participate in mitochondrial protein import. So there are about 30 proteins discovered by the many scientists. Uh, these proteins participate in import of mitochondrial proteins. As researchers learn more about these proteins and how they each reach the respective mitochondrial compartments that utilize them, it becomes evident that there is a multitude of processes that work together in the cell to allow for the mitochondrial biogenesis. Now, about 30 proteins have been discovered by the scientists which participate in the importing of the mitochondrial proteins. That means to allow entry of the mitochondrial precursor proteins, about 30 proteins are involved, and these proteins are encoded by some genes. So some of the proteins will be discussed here, some of them important proteins will be discussed here, and by observing these events, that how these proteins reach to the respective mitochondrial compartments which will be finally utilizing them. So it observed a multitude process, so not a one single process is occurring here, many levels of process are being occurring at the same time to transport this protein, and thus an additional subject of mitochondrial biogenesis has to be studied. So it's a broad mechanism, you can say, that biogenesis of mitochondria is not an easy process. It is done by many proteins to synthesize to import the proteins required for the mitochondrial biogenesis, including the fission of mitochondria, fusion of mitochondria. Okay. Now let us see to the next paragraph. Now it is saying that mitochondria are highly versatile and are able to change their shape through fission and fusion events. Fission means to break apart into two pieces or more than two pieces from the same protein, and fusion can also occur, fusion of two small mitochondria to become a big mitochondria. Definitely, fission is the event where single entity breaking apart, where fusion is the event of two or more entities, here the mitochondria, joining to form a whole part. The process of fission and fusion oppose each other and allow the mitochondrial network to constantly remodel itself. Obviously, this is not done automatically, it is done by some proteins. Some enzymes are responsible for carrying this fission of mitochondria and fusion of mitochondria. And during the process of fission and fusion, it it constantly have to remodel the mitochondria every time. If a stimulus induce change in the balance of fission and fusion of the cell, it would significantly alter the mitochondrial network. So some stimulus, some external agent, if it able to induce a change in the balance of this fission and fusion event within the mitochondria, it can alter the mitochondrial network. For example, increase in the mitochondrial fission would create many fragmented mitochondria, which has been shown useful for eliminating the damaged mitochondria and for creating small mitochondria for efficient transporting to energy demanding areas. So, by balancing the fission of mitochondria and fusion of mitochondria in the cell, it could alter the mitochondrial network. Like if we can increase the mitochondrial fission, that means breaking apart of mitochondria into small entities, it would create many fragmented mitochondria, which has been shown to be useful for eliminating the damaged mitochondria and for creating small mitochondria for efficient transporting to energy demanding areas. So so therefore achieving a balance between these mechanisms allow a cell to have proper organization of its mitochondrial network during biogenesis and may have an important role in muscle adaptation during physiological stress. So there has to be a balance between these mitochondrial fission and fusion, which will help the cell to have proper organization of its mitochondrial network during its biogenesis, and it may have important role in muscle adaptation during physiological stress, because during stress conditions we need more oxygen, we need more energy, and for that we consume more oxygen, and to get that energy more oxidative phosphorylation has to be taken place. Okay, breakdown of that glucose to pyruvate to acyl coenzyme, and then it will be involved in electron transport chain as we have stated before. Okay, so more mitochondria will be needed. So we need a balance between these mechanisms, between this fission of the mitochondrial proteins and fusion of these mitochondrial proteins. And during this time, as I have said that uh, so this process of fission and fusion will oppose each other and allow mitochondrial to constant remodel itself. Okay, so these are diagram where we can see some of the proteins has been named which involved. As simply stated, you can see for the fusion of the mitochondria we need this type of proteins. If we zoom it in, you can see the name of the proteins as stated by MFN1, MFN2 and OPA1. These proteins involved in fusion of the mitochondria from small mitochondria. Well, the main master of the proteins, regulatory protein, is PGC1 alpha, which is the main protein involved in mitochondrial fusion. Now, this is required for increasing the mitochondrial biomass. Okay, this is required for increasing the mitochondrial biomass when we require lots and lots of energy that has to be utilized by the mitochondria. Now, the next thing for the fission, for the fission process, DRP1 protein is involved. This protein is involved during for the fission of the mitochondria to break them into small mitochondria. Now, generally, it is done for removing out the damaged mitochondria. Actually, as I have little bit ago, I have said here it is written. For example, increase in the mitochondrial fission would create many fragmented mitochondria, which has been shown to be useful for eliminating the damaged mitochondria and for creating small mitochondria for efficient transporting to energy demanding areas. So, for we do fission reaction, we do fission of the mitochondria to remove away the damaged mitochondria actually, right? And so that the small mitochondria can able to use their process for create for efficient transporting to energy demanding areas. So a balance between the fusion of the mitochondria and fission of the mitochondria is required. So just go through it. It if MCQ in this term you will get MCQ questions. So generally PGC1 alpha, MFN1, MFN2, OPA1 are the main regulatory proteins for fusion of the mitochondria, and for the fission of the mitochondria we need the protein of DRP1, as you will see now. I will be explaining now. Okay. Now let us see one by one. Slowly I will discuss. Okay, that is required for you in your syllabus about these mitochondrial proteins which are involved in fission, which are involved in fusion. Now let us see. In mammals, mitochondrial fusion and fission both are controlled by GTPase of the dynamine family. Again I repeat, in mammals, mitochondrial fusion and mitochondrial fission, fission means to break apart into smaller fragmented pieces as required to remove away the damaged mitochondria. Both mitochondrial fusion and fission are controlled by GTPase of the dynamine family. GTPase means they require GTP for its activity. Now what is dynamine? As I have said that for mitochondrial fusion and fission, they are controlled by GTPase of the dynamine family. What is dynamine? Dynamine is actually a type of GTPase responsible for endocytosis in the eukaryotic cell. Now you must have remembered about the endocytosis that I have said before. So dynamine is a GTPase which is responsible for endocytosis within the eukaryotic cell. Okay, dynamine is a part of the dynamine superfamily which includes classical dynamines, dynamine like proteins, MX proteins, OPA1, mitofusins, and your GBP. So this much you have to remember that it is a GTPase and it helps in endocytosis in the eukaryotic cell. And what I have said for the mitochondrial biogenesis, that in mammals, mitochondrial fusion and fission are regulated by GTPase of the dynamine family. So for fusion and fission of mitochondrial proteins, we require GTPase activity of the dynamine family. Now, members of the dynamine family are principally involved in cleaving or scission of newly formed vesicles from the membranes of one cellular compartment, and they are targeting to an fusion with another compartment both at cell surface as well as at Golgi apparatus. So generally, those which are the members of the dynamine family, like your here we have mentioned MX proteins, OPA1, mitofusins, GBP. These proteins are principally involved in scission of newly formed vesicles to cleave and form new vesicles from the membranes of one cellular compartment, and they are targeting to and fusion with another cell compartment both at the cell surface or at the Golgi apparatus. Okay, Now here the protein which is involved in mitochondrial fission is the dynamine 1 like protein. Dynamine 1 like protein, as I have mentioned in your figure. Dynamine 1 like protein is involved in fission of the mitochondria. Okay, as it is saying here that dynamine like 1 like protein, it is a GTPase protein, it has a GTPase activity which regulates the mitochondrial fission. What is mitochondrial fission? It is the process where mitochondria divide or segregate, means equally divide into two separate mitochondrial organelles. So this is regulated by dynamine 1 like protein, and dynamine 1 like protein has a GTPase activity. In human, dynamine 1 like protein is typically referred to as dynamine related protein 1 or DRP1, and it is encoded by the gene called DNM1L gene and is a part of the dynamine superfamily DSP family of the proteins. So for the fission, which for the mitochondrial fission, it is regulated by this protein called dynamine 1 like protein. Dynamine 1 like protein has a GTPase activity, and it leads to the separation of the or segregation of the mitochondria into separate mitochondrial organelles. Generally in human Generally in humans, dynamine 1 like protein is referred to as dynamine related protein 1 or DRP1. The gene responsible for this dynamine related protein 1 is your DNM1L gene and is a part of your dynamine superfamily of proteins. So as you can see that for mitochondrial fission, it is regulated by the dynamine 1 like protein which has a GTPase activity. Okay, and in humans it is coded by the DNM1L gene as stated here in the capital letters. Okay, so the process of mitochondrial fission is actually directed by DRP1, which is a member of the cytosolic dynamine family. This protein forms a spiral around the mitochondria and it constricts to break apart both the outer and inner membrane of both the organelle. So, the process of your mitochondrial fission is directed by your DRP1, which I have stated little bit ago. What is the full form of DRP1? Again I repeat, dynamine 1 like protein. So this mitochondrial fission is regulated by DRP1, which is a member of the cytosolic dynamine family. And this protein forms a spiral around the mitochondria and it constricts to break apart both at the outer mitochondrial organelle as well as in the both at the outer membrane and inner membrane of the organelle. That means it will break totally, it will break the outer membrane of the mitochondria as well as the inner membrane of the mitochondria has to be break apart. On the other hand, the process of fusion of the mitochondria is directed by different member anchored dynamine proteins at different levels of mitochondria. As I have said before that fusion, mitochondrial fusion and fission are both controlled by GTPase of the dynamine family. Here for the fission, it is controlled by the DRP1. For the fusion, it is also done by some member anchored dynamine proteins at different levels of the mitochondria. Like for fusion, Fusion at the levels of outer mitochondrial membrane, that means at the outer mitochondrial membrane, fusion is regulated by mfn1 and mfn2, which are also called as mitofusin 1 and mitofusin 2. And fusion at the level of inner mitochondrial membrane is mediated by OPA1. So the inner mitochondrial membrane fusion is mediated by OPA1, whereas the outer mitochondrial membrane fusion is mediated by your mitofusin 1 and mitofusin 2, which are represented as MFN1 and MFN2 respectively. I hope you have understood. For the fusion, it is regulated by many proteins, like fusion of the outer mitochondrial membrane is mediated by MFN1 and MFN2, also called as mitofusin 1 and mitofusin 2. And fusion of inner mitochondrial membrane is mediated by OPA1. Let us see some of the information about this mitofusin 1 and 2 and OPA1, which is also called as dynamine like 120 kilodalton protein mitochondria. So as I have said that mitofusin 1 and mitofusin 2 are responsible for the fusion of the outer mitochondrial membrane. Mitofusin 1 is a protein which is encoded by MFN1 gene in humans. Now this protein is encoded by this gene is a mediator of mitochondrial fusion. This much information I have got. So it is responsible for fusion of the mitochondrial protein, mitochondria. But appropriately, if I say that they are responsible for fusion of outer mitochondrial membrane. Mitofusin 2 is also a fusion protein which is regulating the fusion of the outer mitochondrial membrane. It is encoded by the MFN2 gene. They are all GTPase embedded in the outer mitochondrial membrane, and in mammals MFN1 and MFN2 are essential for the outer mitochondrial membrane fusion. For the inner mitochondrial membrane fusion, we need OPA1 gene, which will encode this protein called dynamine like 120 kilodalton protein, which is encoded by this OPA1 gene. Now, this protein, OPA1 gene, this protein, which is called dynamine like 120 kilodalton protein, it regulates the mitochondrial fusion at the inner mitochondrial membrane and cristae, which is found on the inside of mitochondrial matrix in the inner mitochondrial membrane, and contributes to ATP synthesis and apoptosis and small round mitochondria. So dynamine like 120 kilodalton protein is encoded by the OPA1 gene, and they regulate the mitochondrial fusion and cristae. That means they regulate the inner mitochondrial membrane fusion, and they also contribute to ATP synthesis and apoptosis, which is a programmed cell death, and small round mitochondria. As I have said about the DRP1, that dynamine like protein, they are also a GTPase protein which involve in mitochondrial fission. So dynamine like protein regulates the mitochondrial fission as stated before, which is typically referred to as dynamine related protein 1 or DRP1, and this protein is encoded by DNM1L gene, right? So multiple research studies have been has correlated, and it increases between the mitochondrial respiratory capacity with MFN1, MFN2, DRP1 gene expression after endurance exercise. So multiple research has been done, has been conducted, experiments have been conducted, and a correlation has been found between these proteins MFN1, MFN2, DRP1, which whose expression of the gene increases or decreases during your endurance exercises, during aerobic exercises. Therefore, it is supported that reorganization of the mitochondrial network in muscle cell plays an important role in response to exercise. So there is a supported evidence that reorganization of the mitochondrial network in the muscle cell plays an important role in response to exercise. Okay. Now, what is endurance exercise? I have mentioned here. It is actually to increase our stamina, to increase our flexibility and strength and balance, also called as aerobic exercises. Which it is so scientists have shown that by doing this type of stress exercise, it can increase our mitochondrial biomass, okay, within the cell, and that is mediated by the proteins involved for mitochondrial fusion, right, and distribution also. So fission and fusion both are needed for our maintenance of the mitochondrial number biogenesis. Now the main you can say the master regulator of mitochondrial biogenesis, master regulator of mitochondrial biogenesis is your pgc1 alpha, which is also called as proliferator activated receptor gamma 1 alpha. Why it is called master regulator? Because it is the gene which is at the top of the gene regulation hierarchy. Means it actually the main gene which is regulating the other genes. Okay. So PGC1 alpha, it is the member of peroxisome proliferator activated receptor gamma family of transcriptional co-activators. It is the master regulator of mitochondrial biogenesis. That means this gene, this protein is the head of all the other genes, of all the other proteins. It is mainly involved in the regulation of mitochondrial biogenesis. Now this pgc1 alpha or peroxisome proliferator activated receptor gamma co-activator 1 alpha, it is a protein that in humans is coded by this gene called PPARGC1A gene. So this is the main protein which is regulating the fission and fusion of the mitochondria, and this protein is coded by this gene called PPARGC1A, PPARGC1A gene. It is a transcriptional co-activator. That means it activates the transcriptional factors that are needed for transcription of the mitochondrial genes. How? Let us see. PGC1 alpha is a transcriptional co-activator that regulates the gene involved in the energy metabolism. So again I repeat, PGC1 alpha, it is a transcriptional co-activator. Okay, means it helps to activate the other transcriptional activators which will regulate the genes which are involved in energy metabolism. Actually, this protein called PGC1 alpha, this PGC1 alpha, it first interact with the nuclear receptor called PPAR gamma. This PGC1 alpha is interacting with the nuclear receptor called PPAR gamma, and in turn it will permit the interaction of this protein with the other multiple transcription factors. Simply saying, PGC1 alpha is interacting with what? With the nuclear receptor called PPAR gamma. After interaction with the PPAR gamma, it permits the interaction with the other transcription factors which are involved for the mitochondrial protein formation. This protein can interact and regulate the activity of cyclic AMP response element binding protein and nuclear respiratory factors. Again, I repeat from the beginning. PGC1 alpha. Okay, I have to make small one so that I can able to describe it. PGC1 alpha, it is a co-activator. It is a transcriptional co-activator. This PGC1 alpha is interacting with what? With nuclear receptor called PPAR gamma. Okay, it is interacting with PPAR gamma, which is a nuclear receptor. This nuclear receptor then interact with the other transcription factors, right? Like it regulates the activity of CREB, which is also called as cyclic AMP response element binding protein, and the nuclear respiratory factors. So PGC1 alpha interacting with the nuclear receptor PPAR gamma, which in turn it will cause interaction with multiple transcription factors, and some of them are and two of them like the CREB, called cyclic AMP response element binding protein, and nuclear respiratory factors are also involved in interaction with the nuclear receptor called PPAR gamma. Then NRF, or nuclear respiratory factor, then activate the mitochondrial transcription factors, which directly responsible for the transcribing of nuclear encoded mitochondrial proteins. So, simple thing, PGC1 alpha is interacting with the nuclear receptor PPAR gamma, which in turn it interact with the other transcription factors and other proteins like uh, your CREB, like whose full name is cyclic AMP response element binding protein, and also the nuclear respiratory factors. These two proteins also interact with this nuclear receptor, as it has been mentioned. It has been mentioned here that this protein means nuclear receptor PPAR gamma, as well as your PGC1 alpha. It interacts and regulates the activity of these two proteins called CREB and NRF. The NRF, which is called nuclear respiratory factors, in turn will activate the transcription factors which are needed for the mitochondrial transcription process, mitochondrial gene transcription process, which are found within the nucleus. Better to say, the within the nucleus, the gene responsible for the mitochondrial proteins are going to be transcribed. Okay. This includes both structural and mitochondrial proteins, as well as those involved in mitochondrial DNA transcription, translation, and repair. So again, I repeat, if you have some confusion here, PGC1 alpha full form is what? Peroxisome proliferator activated receptor gamma cofactor 1 alpha. In short, we say PGC1 alpha. It is coded by this gene in humans, PPARGC1A gene. PGC1 alpha, it is a transcriptional co-activator, means it helps to activate the other transcription factors needed for the mitochondrial uh, transcription process. Okay, that some of the genes which will code for mitochondrial proteins, they need to be transcribed in the nuclear genome, right? That is mediated by PGC1 alpha. How? This protein PGC1 alpha interact with the PPAR gamma, which is a nuclear receptor. PPAR gamma in turn will able to interact with the other transcription factors. Okay. PGC1 alpha also interact with the CERB and also with the NRF. What is CERB? Cyclic AMP response element binding protein, and NRF means nuclear respiratory factors. So this protein, PGC1 alpha, it interact with the CERB, cyclic AMP response element binding proteins, and it also interact with the nuclear respiratory factors. After interacting, these nuclear respiratory factors will activate the mitochondrial transcription factors, which are directly responsible for transcribing the nuclear encoded mitochondrial proteins. So the proteins which are actually encoded by the genes which are located in the mitochondria, they has to be transcribed, okay, and translated further, so that they can be transferred to the mitochondrial genome, to the mitochondria. And for this reason, NRF is mainly responsible. NRF is interacted by the PGC1 alpha, which in turn activate these mitochondrial transcription factors called TFAM, which is directly responsible for transcribing the nuclear encoded mitochondrial proteins. So I have said this part so that you can able to recite them. And these include both structural mitochondrial proteins as well as those involving transcription of mitochondrial DNA, those which are involved in translation and repair of mitochondrial DNA. So not only the structural mitochondrial proteins are going to be transcribed here, but the proteins, the genes which are responsible for the transcription of the mitochondrial DNA, like for transcription, what we need transcription factors, we need RNA polymerase, right? It has to be occurred on the mitochondria also, because mitochondria also have their genome. For their transcription to do well, for their translation to do well, we need the proteins, and that proteins are actually here is transcribed by the nuclear genome, which is actually mediated by interact it is done actually with the help of NRF, with the help of nuclear respiratory factor, interacted by the interact by the this protein PGC1 alpha. Okay. Then another protein is there which also plays an important role, which is also called as uh, wait, this one, AMP activated kinase. It is a kinase enzyme which phosphorylates. AMP activated kinase full name is 5' AMP activated protein kinase. In short, we write as AMPK, or 5' adenosine monophosphate activated protein kinase. It plays a important role in mitochondrial biogenesis by simply cause phosphorylating, means it add phosphate group, and activating the PGC1 alpha. As I have mentioned just now, that PGC1 alpha is the main master regulation of mitochondrial biogenesis. So for phosphorylating the PGC1 alpha, and to activate the PGC1 alpha, we need to phosphorylate. We need to have to add phosphate group. Phosphorylation has to be done. That is done by this AMPK, which is called AMP activated kinase, and it is done by upon sensing an energy deficiency in the muscle. So when there is a signal generated, which represents, that signal represents that there must be a energy deficiency in the muscle, so we need we need immediately, we need energy generation, and for this we need more mitochondria to perform their function. And for this reason we need to synthesize more of the mitochondrial proteins, which are most of them located within the nuclear genome. Okay. And at the same time, the mitochondrial synthesis has to be done. Okay. The substrates are also required, on the other hand, that is stored within the livers, within the muscle tissues, right, in the form of glycogen. And in this case, AMP activated kinase, they simply what they do, they phosphorylate the PGC1 alpha upon sensing that energy deficiency within the muscle. PGC1 alpha, then in turn, it is a co-activator, as I have said. It activates, it interacts with this nuclear receptor called PPAR gamma, which in turn will interact with the other transcription factors involved for the mitochondrial gene expression. Then this PGC1 alpha also interact with the CREB, which is called cyclic AMP response element binding protein, and also with the nuclear respiration factor, respiratory factors. Nuclear respiratory factors then in turn will activate the transcription factors, the mitochondrial transcription factors, which are directly responsible for transcribing the mitochondrial proteins encoded by the nuclear genome. So in this way, in a chain reaction, they are doing the biogenesis of the mitochondria. So I hope you have understood what I have today. I have discussed today. Just go through these notes. I am sharing these notes to you now. Okay. And if you have any doubt regarding these notes, you can ask me in the WhatsApp group. And please go through these notes. I am repeatedly telling you. Please go through the video lectures, as I am not able to do the online classes due to not having proper net connection here. Okay. So that's all for today's lecture.